Hello, my name is Paul Zierep. Today I will guide you through the binning tutorial of the Galaxy Train Network, which was developed as part of the Verimax project, of which I am a co-lead, which tries to create a comprehensive workflow and also tutorial for metagenomic assembled genome generation, a crucial part to identify new genomes from metagenomic data. And I hope you enjoy it and you learn something from this, and let's directly dive into the training. So I already opened the training material on top of the Galaxy interface. If you don't know how to set this up, please look into the introduction to Galaxy. And like I said, today we're going through the binning of metagenomic sequences. What are we going to learn today? I hope you will be able to describe what metagenomic binning is, common challenges in metagenomic binning, how you can evaluate the quality of bins. This training is built on top of a metagenomic assembly and is also part of the bigger Verimax generation tutorial. So, what is binning? Binning basically means that you have contigs of which you want to know whether or not they are part of the same organism, and if they are part of the same organism, they should be binned together, basically put together in a file. And there are a lot of different approaches that can be used. Basically what you need information of your sequences, and then you use this information to determine whether or not they are from the same composition or from the same organism. You can use sequence composition based binning. That means you look for specific distinct sequence compositions such as GC content. Coverage-based binning, which is used by most binners. Basically, you say if a sequence has the same coverage in the same samples, then you can assume that it comes from the same organism. And many binners also use multiple of those binning approaches. What are the challenges? High complexity for sure. Metagenomic samples contain DNA from many different organisms, usually. Uneven coverage. Some organisms in metagenomic samples may be more abundant than others. That is very often the case. Then you can also have chimeric sequences and horizontal gene transfer, so there's not always such an easy distinction in real life data sets. And multiple other challenges as well. Strain variation, for example, is particularly challenging because if samples come from the same strain, it's very difficult to determine whether or not they should be binned into different bins. To help bioinformaticians and researchers to bin samples, many different algorithms and tools have been developed. Some of the ones which are quite often used these days are MaxBin, MetaBed, Concrete, but also SemiBin. And for example, one of the latest binners was Combine. And in practice, many of those binners are also used together, and then there's something like a bin refinement step used downstream. So the question for many users is, so many binners, what binners to use? And this is not an easy to answer question. So I can only urge you to look into manuscripts that do benchmarks for those binners, and there's some linked in this training. There you can look which binners perform best for particular biomes, because specific biomes have specific characteristics, and so binners can be different between those biomes, and only comprehensive benchmarks can guide you through this. Another good example of those benchmark manuscripts are the Kami challenges that tried multiple binners on different simulated biomes, and this information can be used to guide you to what is the ideal composition or like this ideal set of binners you want to use. In practice, however, most of the time and also most end-to-end pipelines, they use a set of binners depending on what people are able to set up on their compute cluster or on the machine, or also on what is installed on Galaxy in our case. And then downstream you use something called a bin refiner, which is a tool that takes the output of multiple binners and then tries to generate a non-redundant final bin set. And many benchmarks have shown that this refinement steps can lead to much better bins, higher quality bins. And I think the most famous one, which was used a lot in the past, is Zierep. It has not aged very well. It's not maintained that good anymore. And other tools such as Dust Tool and lately Binet, which claims to be basically the successor of Zierep, slowly replaced Zierep. And are used a lot in common pipelines as bin refining modules. Apart from those bin refiners, there's also Envio, which is special in that regard that it has an interactive interface, so you can manually look at your bins and refine them. So for example, you can check by visual inspection if the abundance coverage makes sense for your bin. So if all the contigs in a bin, for example, have the same abundance information, and then you can manually modify this. We're working on adding Envio to Galaxy, so soon you should be able to use it there as well. We're not there yet to have it fully and nicely integrated. If you want to use it, we're completely not objected. You can create bins with Galaxy and then use Envio downstream in a different environment. So, how is this training organized today? Binning usually is a very time-consuming and resource-consuming step. Machine learning models that are trained on huge amounts of contig data just have this requirement. To make this training here possible, we created a mock data set which is very small. It's not really close to anything real life, but it can be used to run all of those binners. And I mean, that's the important part, that you can generate this training. But all the binners we showed today, and also the complete workflow logic can also be applied to real life large scale data sets. And you can basically start from here and then apply this on your own data. So how do we start with the Galaxy procedure? For this training we need two things. We need to generate two things. We need a FASTA file containing the assembled contigs. So like I said, you should first do the assembly tutorial if you have not done so yet, to look how this can be generated from raw reads. In many cases, Max pipelines use megahit or spades for the assembly step. And then, like we already saw, many binners use abundance information to determine the bins. So you also need the abundance information, and that is contained in a BAM file, which can be generated by mapping the reads of your data set to the assembly contigs. This can be done with Bowtie or, for example, BWA. Can you also generate bins without coverage information? Yes, this is possible. Some binners purely rely on things like composition or GC content and such. Examples are MaxBin and SolidBin. But MaxBin can, for example, also use coverage information if it's available. In general, yes, it's possible, but if you have this information, and I don't see any reason why you should not have it, because usually the people that do the binning, they also have access to the raw reads, then I would always use abundance information, because that has a far better quality of bins downstream. How do we proceed with Galaxy? We do the usual thing, and at this point I assume that you are all familiar with the basic Galaxy logic. So we need to create a new history. I will do this now, so we can click on the main window to go back to Galaxy. Out of the training, I already have history here. I will create a new one now and call that binning. Very simple. I always like to add a tag as well. Let's call it GTA This is very important so you can organize your data in a structured way. If you have created your history, you can also rename it. Like I said, I called it binning. You can call it whatever you want, as long as you're able to find it later when you want to look what you did. Then you can upload the data we need today. So we're going to use the mock config that was created previously with megahit. You click on the upload button and then on past page data, and then you can add your context here and start the upload. Let's have a look what else we need. We also need raw reads. Like I said, we need to create abundance information. I'm also going to copy this part, click on this upload button again, paste and fetch, and upload the raw reads. And then sometimes you need to take a coffee and wait until this is uploaded. So now after a bit of waiting, the data should be uploaded. And now the tutorial tells us to create a paired collection, because this is what the downstream tools usually digest if you have paired read data. To this end, you need to select both read datasets, click on Advanced Build List, and then you need to create a paired dataset. Galaxy needs to know which dataset to put where, so the first element is always the forward, and the second is the reverse. And now Galaxy knows how to match those pairs. And you need to adapt this in case, for example, you have something like F1 or F2 here. Click on Next. We can call this the reads collection. And now we build this collection. And then this should turn into a nice collection where your data is stored together. So after you create this collection, we can start with our first tool. Like we said in the introduction, we need to create a mapping file that maps those raw reads to the assembly or to the context. And in this tutorial, we're going to use bowtie. BWR is also fine. But in the bigger scale of, for example, the Fairymax project, where we have a comprehensive workflow that goes all the way from raw reads to the max, we use bowtie as the default mapping tool. So I would recommend to use that in all steps as well, because it makes downstream easier because the file format is already clear. Let's see what parameters we need to set. So the input will be paired end reads, and we're using our raw reads data input datasets. And we don't want to use a built-in genome index, but we want to build from history because we have the data available. And I think apart from these options, the only thing we can set here is save bowtie mapping statistics to history. I mean, it makes sense to have a look at this. So let's click this. And apart from this, all parameters can stay default. So now we can run this tool. That should start a job. And let's wait until this one is finished. After the mapping is done, you will also need to sort this BAM file. This is a normal step before downstream processing. So let's click on SAM tool sort. And you need this also for many other steps. And we will sort it by coordinate, meaning the order of the mapping on the context coordinates. And let's run this tool as well. While this is run, we can have a quick look on the mapping statistics. So here you can see that of the paired reads, 90% aligned only 0 times, means 90% did not align. 90 did not align at all. And 98% aligned exactly 1 time. So for bigger projects, you should always make sure that the vast majority of reads align. If not, there's something wrong, because I mean the assembly is created from those reads. So, after the BAM file is sorted, we can now start to actually bin our contigs. Once again, there are multiple challenges, but the overall aim is clear. We have our contigs and we want to put them in bins that belong to the same organism. This tutorial is a choose your own tutorial. That means you can basically follow the path now for multiple different binners. I will only look into metabed, but you can feel free to look into how the binning is done with max bin, semi bin, concat, not combine. Important note to combine. We added combine as a binner here because it's a very powerful tool. It has been shown in multiple benchmarks that it's very performant, but it has a downside. It cannot really work with small data sets. In real life data sets, this is not really a big problem, because most of the time you have rather big data sets and Common Bin can work with this. Don't run it on the data sets here, it will fail. It's a tool constraint, has nothing to do with Galaxy. But if you want to use it on your own data set, that's a valid approach. It will probably create one of the highest numbers of high quality bins. We are only going to look into metabed, but like I said, you can feel free to also look into the other binning algorithms. So let's look into metabed. Metabed uses a combination of tetranucleotide frequencies, so read properties, coverage depth, and also read linkage to create contigs. Like many other binners, it uses multiple. It can scale to large scale data sets. And is widely used by the community. It needs another pre-processing step. You need to calculate the contig depth, so it needs like a tabular file with the contig depth, and you can process this based on your SAM tools. We're going to use this tool, calculate contig depth one by one. Let's have a look, and we're going to use the SAM tools sort output to generate this. This needs to be added here, and then you can run this tool. So, after we run this tool, you should get a file like this, which now is a tabular file that shows you the total average depth for each contig. And now, with this tool, we can run metabed. Click on metabed, and then use the contigs as input, and you see you don't necessarily need to add the abundance information. So apparently I have created this training based on older training. We didn't use abundance information. If you want, you can use abundance information just by clicking on yes coverage file, and then you can use this file as input here, and then this will use the abundance information to generate the bins. I will not use this option just to be consistent with the training. Yeah. There's one other issue here with this training. It says that you should save cluster membership as matrix format. This is something that you can do and is useful for some downstream tools. In our case, we don't need it, so we will set metabed to not use this option. And then when you run this job, you should get an output like this. And the important information here is found in the bin sequences. So here we will find bins, and we have generated one bin which contains 54 sequences. There are also other data sets which are created sometimes when you run metabed, depending whether or not you set these options, but the most important part is the final bin set of the genome. The question was how many bins were produced by metabed for our sample. There's only one bin, this is still correct. How many contigs are in the bins? It says 52 in our training. I think the metabed version was updated since the last training. so that can change slightly. Also because internally MetaBed has a non-deterministic approach. So you can see in our case we found some more bins, and that's one of the caveats of metagenomic binning is that between algorithms some slight difference can often happen, also depending on the deterministic method which underlies this process. I will in this tutorial not go into the details of each of the binners. You can do it for yourself. Like we have seen, all of the binners can be investigated with this training, and feel free to execute each of them on your binning, on your reads, or on your context, better say. What we would like to see here now is how the results of multiple binners can be refined using one binning refiner called Binnet. You can also use das tool, which is also included in Galaxy. Since we will not create all the bins ourselves, we will just import these bins created by those binners, which are already generated here. So you can copy these links and then upload the data using the upload tool, test fetch, and then start the upload. For downstream processing, we need the bins from each binner in a separate collection. So you can see here, for example, that Concat produces a lot of bins. That's typical for Concat, and some of them need to be removed by the downstream processing, refinement process. To correct this collection, and you can already do this while the files are uploaded, you once again need to select all of the bins that needs to go into the same collection. So in this case I collect all of the Concat bins, set advanced build list, a flat list, and we call this now the concoct set. And you need to repeat this for all of your bins. So now I also have downloaded all of the bins and formatted them into a collection. And to make life a little bit easier, I also added the information of which binner this comes from, using the hash logic. So this hash tag will also propagate downstream now. For the binners for the bin refining tool to process this data now, we need to use the convert genome bins in FASTA format to generate basically a bin table. And we can use this tool for that. So you can click on this tool and then select one of your binners. I will start with semi-bin, and then you run this tool. And we need to apply this to every bin set, and this way this tag is very nice, so you cannot lose track. So we apply this to MetaBed as well. We apply it to MaxBin, and once again to Concoct. And we need to wait a little bit until this is finished. And when those jobs have finished, you should see that each of the set of bins was created in such a binning tabular file. So for example, let's look at semi-bin, which doesn't show ready yet. For example, for MetaBed, you can see now that this is just a table where for each contact you have the assignment of the bin. So here are all the bins put together. For example, looking at Concoct, you will see that it created multiple, it has a binning table with multiple files. Most of them are, however, collected in one bin. After these jobs, we need to put those contig bin mapping files into one collection, because Binet downstream needs one collection where all the bin assignments of your binners are put together. So we can use this build list tool, and we have to insert multiple data sets. So the one from Concord, from MaxBin, from Metabed, and from SemiBin. We're using the index, and this tool will now run instantly because it's a collection tool. And you will see now all the labels are here, so they are all included in this collection. And when you click on this, you will see one table where you see that all the individual files before are put together in one collection. And this is now what you can use for Binet. So we click on Binet. Like I said, one of the latest bin refiners. Now we have to provide the input table. This is this one. Then also input config table, build this input config file. And for the input config file, we need to use the context faster file. It can also use output from Prodigy, but we're not going to use this. And you can also determine what is the minimum completeness of this tool. We will use the default parameters. In practice, you can also decide whether or not you want to weight contamination or completeness more in the refinement step. We use the cache database, and now we can also run this tool to see to start the refinement step.
So how does the refinement work? So there's a question here: how many bins are left after refinement? And if you successfully executed Binet, you should see that you have 2 bins left. So most of the individual bins from the binners were combined into one bin. There's still one single config bin which could not be correctly assigned, but still Binet did a good job to consolidate this. And so now we created bins, and we also created refined bins. And the usual downstream step of this now would be to check the quality of the bins and then decide whether or not you want to use them downstream for max, for example, max taxonomic or genome annotation tasks, or also publish them. That would be the final goal of a max workflow in many cases. And the de facto standard tool to use for bin quality investigation would be checkm. There's checkm1, where usually the lineage workflow is used to estimate completeness and contamination of the bins. By now, there is also there's also checkm2 available. Checkm2 uses a more machine learning based approach, which apparently is able to also determine completeness for genomes which are rather rare. checkm1 is still used beside checkm2 since it has a more heuristic approach looking at conserved genes. We're going to use checkm1. You can also use checkm2 if you want to investigate the quality of our bins. Therefore, you need to run checkm lineage workflow on any of the folders that contain the bins. So that means we have to run checkm lineage workflow. And we need to run that on our refined bins, but also on the bins of each of the binners. I'm going to show it as example for the refined bins. You can also, like I said, run it for all our other bin sets. We're going to use default parameters, and no, I think we don't need any additional information here. And then you click on run. All right, And when you have executed checkm and on all your bins, you should see something like this for each of your bin sets, where the important part are the bin statistics. Let's have a look, for example, on the bins of a semi-bin. Then you should see a file for your bin you see the marker lineage, so this was a bacterial source, and you see contamination and completeness. Since this was mock data, it's a very small incomplete genome. In real life, you should aim for something like 75 or 80% for genomes that you want to report. And you can compare this for all the other examples as well. For example, Concord, as we see before, there are multiple sets, and only one of them has a completeness. All the other ones have close to zero completeness. And you find in the training a comprehensive list of all your reports for metabed, semibin, maxbin, and concat. And you can see that all those bins were nicely consolidated into a final bin set from Binnet, which has now a completeness of 15.67, which is on par with the best bins found from our binners, showing the quality of Binnet. In practice, you would now also filter out all the bins with a low quality. So where do we go from here? So practically this tutorial is finished now. You've seen to use multiple binners and also how to execute bin refinement. And you can also explore multiple other options available in Galaxy. One downstream process that is done often from now would be dereplication, meaning you might be able to find a lot of bins in your different samples, but they all might be on the species level belong to the same species. And what is done to consolidate data of complete studies usually is to dereplicate, for example, on species level with a similarity of 95%, and then report the best max found for your study. And for example, since there is often a discussion whether or not max found are reliable, because of course by this computational process there can be chimeric artifacts happening. I would say the more similar max you find in a study or even cross study, the more reliable your max are. For example, if you find 10 max that are on the species level identical in 10 different samples, that this is very likely to be a true mac, which also has a existence, so to say, in the biological world. And for example, if you look at the main max workflow, which is also available in GTN, you will see all the way the process all the way down to dereplication, which I can highly recommend you to follow up after this training. So in conclusion, you've seen how to bin your assembly using Galaxy. You saw one binner, but you can also from here on try other binners. In practice, it always makes sense to try different binners, try different version of binners, try especially in relation to your biomes, then try bin refinement and investigate the quality of your bins, and that should really help you to find new bins and also find new max for your biome using Galaxy. Thank you very much for doing this tutorial. Good luck with your own analysis tasks, and as always, feel free to reach out to the Galaxy community if you have any problems with your analysis. Have a great day.